What I've been working on is a program. Actually, it gets at the model definition language and interpreter called Antimony. I called it Antimony because antimony symbol is SB. See, there you go. The basic idea is to make a human readable and writable model definition language. So this is an example basic thing here. You can imagine typing up something like this when you're defining a model. And then let's see here, probably this one. And right now it's a command line tool, but it's packaged as a library. So the idea is that you could use it as a plugin for whatever. Demo.txt, and it reads it in. And this is a little. This is just something that sort of spews out the information that it gathered, and you can use the API to get at all this information. So as you can see from the original model on the left, let me try to wrong. There we go. It's now sort of gathered in all the symbols. Did I turn something off? And knows something about what they all are. So like you used A, B, C, D, and E in reactions, so it knows those are species. It never saw K0 or K1 anywhere outside of a formula, so it knows those are probably just constants. It named the reactions, so if you needed to get handles on them, those are sort of the default things. And then you can get things like the stoichiometry matrix out of that, and then the reaction rates for those reactions. So this is at this level, it's sort of very similar to a program that Herbert wrote a while back called Jarnac, which again is you type in models and it knows what you're doing, and then you can export them to a simulator. So the advantage of Antimony, as to differ it from Jarnac, is that you can use it modularly, and you can include different models. So demo.txt is the one we just did. This is demo 2, and then we're going to import it into this. So and you can also import SBML files. Oscilli.xml is XML. So if we look at just the SBML model, it does the same thing for that as it did for the one we defined. So this would be like the Antimony output for that. And again, here's all the reactions, all the variables that it pulled out of that file, the reactions, the stoichiometry matrix for the thing that was all defined in SBML. And then it uses libSBML to get this out and convert it into Antimony data structures. So now, if we run this, you can see that now we have one huge, there we go. So now we have a list of all the species and reactions and everything from both those models, because we've pulled them in as M1 and M2. And then you can manipulate them, like I've done at the bottom there. You can say, in this version, I want S1 to start at 4.3 instead of whatever it was before, and I want A to start at 3.3, and it was undefined in the previous versions. And now so you can use this sort of thing to like have different starting conditions under the same model and easily keep track of them all. And I should point out that libSB or SBML itself is moving towards this sort of modular architecture and has a very almost sort of independently invented modular thing where you take the basic model and then you say, I'm going to change it in this way and this way. I'm going to add this. I'm going to take this out, which should be making design things more easy. And then you can also imagine a visual tool that takes all the modules that you say and sort of packages them up together, and you could view them and in Tinkercell is probably going to do that sort of thing, where you view your module as a black box, or you can expand it and see all the things that are inside it. And just for fun, I'll show you a little thing that a slightly different format that's not in SVML. But this is a special format for defining DNA strands. And I'll just wave my hands around here. So you can imagine here you have promoters and RBS and genes and a stop codon, and then you define them all this way. And there's this, I'm not sure exactly how relevant this will be, but just in case, the ellipses is like anything upstream of RBS, it sort of automatically inherits. And we can imagine a ribosome binding site that multiplies whatever rate it gets in by 0.3. And then, or a different RBS that multiplies whatever rate it gets in by increases it. And then G1 and G2 are genes. They are actually reactions where you have nothing going to a protein. And then stop will stop the thing. So if I were to attach anything downstream of stop, everything sort of automatically inherits whatever I had stuck upstream of it. So you could attach something downstream of stop, and it would inherit 0 now. I won't demo that because there's a bug in the program and something's going wrong. But anyway, but the same idea would happen. You'd get a bunch of things in your program, and you export it as a stoichiometry matrix, or for your visualization tool, or whatever else you wanted. Yeah.
Is it possible to replace those RBS constants with a function of perhaps another variable or something?
Yeah, exactly. Yeah, you can put. So yeah, you could go. Yeah, whatever. Times K0 or K1 or whatever, or you could also say, like what this ends up doing is this, since I know that I put RBS2 right downstream of P2, this actually is P2 times 1.5, or I could just code that explicitly in it or something. Similarly here, I can define the reaction rate of G1 as RBS1. And that's the default. Or I could say that maybe, like, you can imagine something where G3 like this, and G3 makes protein C at say it has unusual codons or something, so the rate is slowed. So you say but it doesn't make that inherit from the it doesn't downstream of G3 would still inherit what's upstream of it, but it itself has a lower rate because it has unusual codons or whatever. So that's the basic idea. Are there any questions? Yeah.
I think one thing I may not have come across is
okay.
It's very hierarchical. So you can build up very complex pathways from lower model definition to combine them together.
Right.
That's the main emphasis. So from the HTML point of view, it was because we wanted libraries of models in the system biology.
Yeah.
Combine models together in an easy syntax.
Right. So you could do something like and with M2 is another copy basically of two genes. Right? And then you'd have separate copies of those, and then you could define them. You could say, and then you could say the genes are actually the same, or you can so something like we haven't quite defined a syntax for this, but like and 1.g2 is m2.g1 or something, or maybe an equal sign or something, to connect your different modules to say this is actually this. So then maybe I only want one stop codon. You say like there's only one stop or formula in the whole thing, so I don't have to keep saying stop 1 is 0, stop 2 is 0, stop 3 is 0, things like that.
So just to comment again, not to start again applying the VLSI CAD stuff to this, but just saying this is very similar to something called, there's two hardware description languages, VHDL and Verilog. And there's a whole industry called the IP industry of Verilog and VHDL, where companies actually just make this code, and they sell it, and it synthesizes the hardware. And the reason you can do that is because of this kind of hierarchy and this modularity, that I can write my own module. I have it protected as that's my IP, and then I sell that to someone. I'm not advocating that model, but I'm saying that modularity, the fact that you have this
is a big deal, yeah.
is extremely powerful, and another reason that CAD for VLSI works. So again, I've just seen this now, but I think that's again a powerful thing to maintain.
Yeah. The whole one of the main ideas behind doing this whole project was to incorporate modularity into the model definition language field, I guess. So, any other questions? I'm going to turn it over to.
This is a relatively new software. I've just been working on it for about 2 or 3 months. Let's see. So, to summarize, essentially what it is it's systems biology provides all these. There's a lot of softwares that you can simulate. You can build networks, visually simulate them, analyze them, and things like that. But when you move those to synthetic biology, there's some needs that are not met. For example, one thing, for example, is this software supports this tree of parts, and this tree of parts is loaded from an XML file. And each part, for example, I can put down this gene, and then each part has a set of attributes, such as this one has length, pops, sequence, and things like that. And those are all loaded from an XML file. So you can imagine someday, hopefully, if there's a database of parts with information about sequence, well, there is information currently on sequence, but suppose there's kinetic information and some other information that characterize parts, then this program can automatically load in that tree along with the attributes. So like RBS might have a particular attribute perhaps in the future where it describes the strength at which it upregulates translation. So that's one part where you can, it combines the simulation capabilities with database of parts so that you can actually, and once there is a database like that, you'll be able to just right-click and say, load all the attributes from that database. So you'll actually be able to use existing parts in your simulation. So that's one part. The other part is, suppose you make a little simple network like that. Let's see, I'm changing the concentrations here of those species. So basically this visual side, so I'll talk about, the basic idea behind, so the program is called Tinkercell, like Tinkerbell. The basic idea was to allow as much flexibility as possible as far as the way you simulate the model or analyze the model. And the reason for that is there has been some interesting works that show that sometimes you need to simulate a model in a lot of detail in order to match the actual biological data. For example, a simple example is well, I'll go back to my first step. So if you have a simple system like that where you have promoter, RBS, gene. So this reaction represents like a POPs flow. So you have, this shows sort of, you have, the promoter generating this POPs, and the POPs is flowing from D to B and from B to A and A to C. And there's some works that show where you might need to model this explicitly. You might need to model the actual polymerase moving through the DNA in order to match the actual data accurately. So the intention was to leave the modeling part as open as possible, so that suppose in the future, we don't know exactly what sort of modeling would exactly be needed to model these things accurately for engineering biology. So what happens is the visual part is just a set of reactions and attributes, and all the analysis and simulation is done by C functions. So you can actually write just C functions that will do things. For example, you can do this thing such as I'll just say this thing called object x equals model dot find a, and then I can say model dot select a. So select x and run that. So it selected that. Now that's not a big deal, but it is a big deal because this is actually a C function. It's being compiled and run. So you can do a lot of things. Basically, you get a lot of these functions that allow your C function. The Tinker Cell provides a lot of functions that can be called from C. That way, all the analysis, all the simulation is actually done by C. And therefore,
The point is speed at that point.
Yeah, the point is not just speed and also flexibility, because you can basically do all the analysis. You can do anything that you can program, but at the same time, it allows you to build your model visually. So it adds the power of programming with the ease of visually building things. So, for example, there's this simulation there's a simulation that is actually done by C function. So I don't know if you noticed that thing that popped up there. It's actually compiling my model into a C function and then running that function. And it's all multi-threaded, so you can run multiple simulations at the same time. While you're running a simulator, you can work on your program. And it's compiled. So there is a time for compiling the code, but after that, it should be very fast. It should be faster than traditional simulators. And there's a stochastic simulator in there also. I'll move this here. And you saw two windows. One was compiling and the other was running. And you see the compile took actually longer than running. But as your model gets complicated, it'll get much faster. So I'll demonstrate, one of the things that you cannot do in most model building softwares because it doesn't support, the power of programming, and that is, let's see. So I have, also, just like there's a tree of parts, there's also a tree of connections. For example, there's a simple reactions, and you get different reactions here, like 1 to 2, 2 to 3, whatever. And this is a transcription regulation. So I'll say both of these regulate this promoter. Now I can view the stoichiometry matrix of all the whole system. So this shows me the stoichiometry matrix, which means that's the rates. I'll just point it here. These are the reactions that are in the system, and these are the rates at which they're happening. So if you would notice, I have these two reactions, these binding reactions. So F and E are binding to D. It doesn't really make sense so the stoichiometry doesn't really reflect the binding reactions. So I wrote this C function that actually, I click this button, it runs a C function. What it did is it changed the stoichiometry. So please ask questions if this is confusing. So, before the stoichiometry was just 2 by 2. What that C function did is it made both of those functions it generated the stoichiometry that represents the full binding and unbinding reactions. For example, in actual biology, what's happening is if you have this regulatory region, E can bind to that regulatory region and unbind. F can bind and unbind. E can bind, then F can bind. So you have like 2 to the power of 3 different reactions going on. And there's no way you'll construct that by hand, especially if you have like 3 things. There's no way you'll construct that in any way other than programming it. So this software, allows you to add the power of programming as well as, build it visually. And then you should be able to simulate that. Let me change a few kinetics so that the simulation looks better. J0, change that to 20 or something. Oh, and let me degrade these things. That degrades, this degrades, and then let's see. Okay, so that's not, you can't see the delay, but there is a small delay. Yeah, it's not very clear. Basically, there's a delay between, so, like I mentioned, there's this flow. D is getting activated, and there's this flow from D to B, B to A. So there are actually 2 lines here. A gets activated. The pops of A, the polymerase reaches A after B. And you can do complicated simulations, like you can take the length into account. You can add the length of V into the rate and things like that. So a lot of flexibility in the modeling, and accommodates database access, and nice little GUI features like that. And that basically summarizes it, I think. And there's little things like you can search for sequences here. Let's say this particular gene has a particular sequence, like something like that. Then you can search for that here. Oops, GT. Forgot the sequence that I put in. AAGTT. AAGTT. And it'll highlight the part. You can even do regular expressions. Little nice things like that. So, you can imagine a system with a lot of genes and things like that. You can search for sequences. You can add a C function that might blast to do whatever. And there's a plugin interface. So all these things, all these windows you see, that the top bar, all these things, they're all plugins. Without plugins, the software doesn't have anything. It's just like a blank slate. So the plugins do everything. And on top of plugins, you have the C interface that can do all the analysis. And I tried to make it look good. And that's all. So one thing I would like to address is I think that some of the features like this, tree of parts, being able to simulate from that, it's sort of ahead of its time. And because we don't really know what all information we need to accurately simulate the biological system, a synthetic system. And there is no database right now of all the kinetics and things like that. So a lot of things that this program can do, I think, are somewhat ahead of the time. But I don't think that's necessarily a bad thing, because it sort of pushes that, it promotes the thing that we need to have detailed models. We need to have kinetic information in databases. So if a tool is ready for things like that, hopefully somebody will take the effort, or we will take the effort, hopefully, to build that database. And yeah, it has the, sorry, I keep going. So it has, like modules, but I haven't worked on this. It's just a box right now, but someday, someday you'll be able to put boxes around that, and it'll mean something. Yeah. And right now you can put these things in there, it'll just move along, but it doesn't really mean anything kinetically. And that's all. Hopefully that was clear.